Because I didn't set my timer, my stopwatch, that last lecture 2 may be split into 2 and 3. I'll just have to see, and I number them depending on how the camera breaks them up. But this will either be lecture 3 from the PowerPoint or lecture 4. Okay, now we had talked about pleiotropy. We had talked about epistasis. We had talked about continuous variation. Oh, and of course, earlier we talked about complete dominance. Now, there's incomplete dominance. And what we have is the heterozygote has a unique phenotype. So we have 3 genotypes, if we're looking at just one allele or one gene, and its alleles. 3 genotypes, like we do with complete dominance, but we also have 3 phenotypes. Okay, and I'll do an example of one of those for you when I get to the board work. But an example in humans, hair. Curly hair is incompletely dominant over straight hair in humans. So a person with curly hair is homozygous dominant. A person with wavy hair is heterozygous. And a person with straight hair is homozygous recessive. Okay? We'll do some more with that on the board. Then finally, there's codominance. So this is where there's a gene, there are 2 alleles, and both of them are expressed. Instead of with complete dominance, the dominant allele hiding the recessive allele completely, so that big T plants are tall. They're not medium, they're tall. Just like big T. Here we have a cow, and this brown is completely dominant over white. This reddish color is completely dominant over white. But the brown and the reddish colors are codominant. They both show up here with this particular cow. For us, and what I'm going to focus on when we do a Punnett square, will be hemoglobin. Hemoglobin A is completely dominant over hemoglobin O. Blood type, sorry. Back that truck up. Blood type A is completely dominant over blood type O. Blood type B is completely dominant over blood type O. But blood type A is codominant with blood type B. Another example in humans. Hemoglobin A is codominant with hemoglobin S. Okay? So hemoglobin A and B, if there are alleles for those, if a person has an allele for A and an allele for B, their blood type will be AB. We'll talk about that some more. Okay, so this will bring us to human genetics, and there's so much more. But I have some examples, and I have 3 categories for us to look at, and then examples in each of those. Autosomal recessive inheritance. With autosomal recessive inheritance, the problem allele, because we're mostly it's not always problems, but we're going to look at some diseases. So problem allele is a recessive allele. Okay? So that an individual would not express or have a phenotype associated with that allele unless they were homozygous recessive. That's why it's called autosomal, part of this, the recessive part of autosomal recessive. The other part, the autosomal part, is that it's not on a sex chromosome. So the gene is, for us, on chromosome, either chromosome 1, 2, 3, continuing down to 22, okay? So it's on one of those 22 chromosomes that are autosomes. And the issue, and it's not always a disease. Albinism isn't a disease, it's a condition, but albinism is another one. If for a person to be an albino, they are homozygous recessive for skin pigment. Okay. One of the examples I'm giving you is cystic fibrosis. Cystic fibrosis is a genetic disorder. Cystic fibrosis itself is a disease that is associated with a problem protein. And from looking at transcription and translation, we know that's what the DNA does. That's what the genes do. So there's a normal allele for a chloride transport protein. So most of us, in fact, we've got that. We've got that. In fact, we probably have two normal alleles. We're probably homozygous dominant. And all we make is the good protein, and we don't have cystic fibrosis. Okay? People who have two alleles for the bad protein cannot move chloride. It's a chloride transport protein, so they cannot move chloride appropriately into and out of their cells. And the secondary effect or result is that mucus builds up, and that's the problem. Cystic fibrosis is a disease involving the buildup of mucus in particularly the lungs, the liver, and the pancreas. So this mucus builds up and it causes problems. Look, you don't want a lot of mucus in your lungs. That impairs your ability to breathe, to get enough oxygen to your cells for cellular respiration. Okay, because if it's wet, if it's in liquid, or not really liquid, but mucus, so jelly-like stuff, that gas exchange is not occurring. So cystic fibrosis has historically been a very extreme disease, and when I was in my 20s, the person who had lived the longest with cystic fibrosis, so we're talking and I'm talking early 20s, so in the 80s, was still a teenager. That was the oldest person to survive. That's how old they got. Most of them died as younger teenagers. Thankfully, we have much better treatments. So people with cystic fibrosis can live longer. And potentially with germline gene therapy, there might be the possibility to eradicate cystic fibrosis. Because cystic fibrosis, even though it doesn't kill as rapidly as it used to, it is a very progressive and awful disease. One of the things that this is apparent, the child has cystic fibrosis, and each night they have to loosen the mucus in the lungs. So they do this thing called thumping, where they're not hitting them to hurt them, and they're not really hurting them, but they're having to, a little bit more forceful patting on the chest and the back to loosen this mucus so they can continue to breathe. Okay? This is more common in Caucasians or whites. Why? Well, the mutation, that's the evolution part, arose in someone that was white. And because up until more recently, humans didn't necessarily have a lot of babies with someone that was of a different race. Okay. Phenylketonuria. there's a possibility that someone in this class is a phenylketonuria. You can't tell by looking at them. This is a physiological phenotype. Actually, you can't tell just by looking at someone that they have cystic fibrosis either. So these are physiological things. So, again, there's a normal allele for an enzyme that metabolizes the amino acid phenylalanine. And then there's an abnormal allele for the enzyme that metabolizes the amino acid phenylalanine. People who are homozygous dominant and heterozygous are able to produce this enzyme that metabolizes phenylalanine properly. People who only have the mutant allele, the mutated allele, that makes an enzyme, but it doesn't metabolize, it doesn't fit, for example, the phenylalanine, its substrate, so it doesn't get metabolized, so it builds up. When it builds up in a child over time, that causes, and hopefully I took that out of there, it causes brain damage and thus the development of delays, okay? What we call developmental delays. I don't want to use the R word. Okay, now the cool thing about this is babies can be screened. They can be screened for other diseases. That's the last slide I have to show you. They can be screened for other diseases so that you can find out if these children have phenylketonuria, if they're homozygous recessive, and there's a fairly simple solution. You make sure that they don't have phenylalanine in their diets. In fact, pick up a Diet Coke or a Diet Pepsi and read the label. It says, warning, phenylketonuria. This product contains phenylalanine. You can see if I'm correct on that. Okay, newborn screening. Oh, yeah, I put that there. Okay. I took out let me go back. I took out sickle cell, but because technically it's not autosomal recessive, but the pattern holds, and I'll show that when I do a Punnett square for you, that someone has to be homozygous for hemoglobin S to have the full-blown disease of sickle cell anemia, okay? So over time, and you can look at a timeline, there was a time when only the newborn screening was for phenylketonuria, but as time has gone by and they've come up with more testing, they can screen children for some of these disorders that if they know about, and these aren't, I said children, I'm talking newborns, they can screen them, they can make changes, give them medications, keep certain things out of their diet, and wow, these children do not end up with these diseases. Now, you can't prevent sickle cell anemia right now, but again, that may be, and oh wait, let me back that up. If you look online, in fact, it's surprising to me, it's somatic cell gene therapy, in which I find very confusing, but apparently they've done some somatic cell gene therapy on a few people with sickle cell anemia, and they claim that they have cured them. That's cool. I don't know I guess it works because they get into the bone marrow where there are cells called blast, and they replace their blast cells that make their red blood cells. So without having to go to germline therapy, they're able to cure sickle cell anemia. Woohoo! Because that's not a good one to have either. So there's sickle cell anemia. I mentioned it was unique. This one's more common in people from Africa who happen to be black. And that's because that's where the mutation arose. And again, over time. Now, we see a lot of now it doesn't matter. People have babies with people of other skin colors. Great. And that's wonderful. In fact, I hope that we can get rid of racism this way. But, oops, I need to stop pretty soon. Until that started happening big time, we wouldn't see people with apparently white skin who might have hemoglobin S. But now there might be people who are white as far as just their outward phenotype who could be carrying the hemoglobin S allele. But it is still way more common in people who are black. Okay, I need to stop again.